Bacteria can be grown in the lab on an agar plate or in a nutrient broth. The culture medium contains carbohydrates, minerals, proteins, and vitamins. It is important that bacteria should not be grown at temperatures above 25 degrees Celsius in a school lab. This is to prevent harmful pathogens to grow, as they are more likely to grow at this temperature. You need to be able to investigate the effects of antibiotics on bacterial growth. So we're going to place paper discs in different antibiotics, so antibiotic A and antibiotic B, and also control. Have one disc that is not placed in an antibiotic for a control, and that's this one here. We're then going to place these into the agar plate that contains bacteria. So the blue is the bacteria. The antibiotic will diffuse into the agar jelly, and it will kill bacteria that are not resistant to it. A clear area will be left where the bacteria have died, and this is called the inhibition zone. And we can see the clear area here. We're then going to incubate the plate at 25 degrees Celsius for 48 hours. The more effective the antibiotic, the larger the zone of inhibition, as it's killed more bacteria. A has a larger zone of inhibition than B, so it is a more effective antibiotic. A control is important to ensure that the results are not down to another factor, such as the paper. You need to be able to describe how to prepare an uncontaminated bacterial culture. So we should sterilize the petri dish and culture medium before using them. You can do this by heating them to a high temperature. This is to kill unwanted microorganisms. You're also going to sterilize the inoculating loop. You can pass it through an open or hot flame. And also the lid in the petri dish should be lightly taped together to stop microorganisms from getting in. You're also going to store petri dishes upside down. This will stop drops of condensation falling on the agar surface. You can measure the inhibition zone, and remember that is here, so the area where the bacteria have been killed. You're going to use a ruler to work out the diameter of the zone. So here's using a ruler to work out the diameter. Half this to get the radius, and then use the formula equation. The area of the inhibition zone is equal to pi r squared. You have now learnt about culturing microorganisms. You will find a summary of this information below. Make sure you test yourself using the quiz on reviseitright.com. Why are we the best revision website out there? Well, because we have it all in one place. Revision notes, summary videos, longer, more detailed videos for topics you're struggling on, exam question walkthrough videos where qualified teachers take you through the perfect answers to exam questions, worksheets, quizzes, computer marked exam questions, interactive flashcards, forums, the ability to keep track on where you are using our tick list, and so much more. But that's not all. We run at least 3 live lessons a week, and closer to exam time, this will be more, where you can tune into a qualified teacher teaching lots of different topics each week. The lessons will be interactive, and we go through exam questions and quick quizzes to ensure understanding as you go. We also run drop-in sessions where you can drop in, ask a tutor anything live, get the answer that you need, and leave. These are included in our silver and gold membership. This means that for an extra £10 a month, you can access at least 12 hours of lessons a month. That's under £1 an hour, which is incredibly cheap. Still not convinced? Try us completely free for 7 days and cancel anytime, completely free of charge. We offer this because we're convinced that you'll love us. So try us now.